Hello, friend. Welcome to Science for Sleep. Cinnamon creates a feeling of warmth in the mouth, even though no actual heat is present. The temperature of the spice does not rise. The air around you does not change. Yet the body registers something that feels gently warming. This sensation comes from a molecule called cinnamaldehyde, the main aromatic compound in cinnamon. When it touches the lining of the mouth, it activates sensory receptors that normally respond to warmth and irritation. The nervous system interprets this activation as heat, even though no flame, no steam, and no rising temperature exists. It is a small chemical signal, translated into a larger perception. The body is constantly interpreting signals like this. Receptors detect pressure, light, scent, and texture. They convert physical input into electrical messages. Cinnamon simply participates in that same system. Cinnamaldehyde binds to specific channels on nerve endings, including receptors involved in sensing temperature changes. The result is a soft illusion of warmth, not burning like chili peppers, which rely on capsaicin and pain receptors, but something milder and rounder, a quiet suggestion of heat. If you enjoy these quiet journeys, you're welcome to subscribe, and if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find their way to this calm corner of the world, one sleepy traveler at a time. The quiet continues here, just as it is. Long before cinnamon is powder in a jar, it is bark. It grows on evergreen trees in the genus Cinnamomum, native to warm, humid regions. The part used as spice is the inner bark. Farmers gently cut the outer layer away and peel thin sheets from the inside. When these strips are left to dry in open air, something simple but beautiful happens. As moisture evaporates, the bark contracts unevenly. The inner surface shrinks at a different rate than the outer layer. This difference in tension causes the bark to curl inward. It rolls into itself, forming the familiar quills that stack inside glass jars. The curl is not shaped by hand. It is physics. Water leaves the plant fibers. The fibers tighten. The structure bends. Over time, the color deepens from pale tan to warm brown, as oxidation slowly alters the surface compounds. The scent becomes more concentrated as volatile oils remain trapped within the rolled layers. Even after drying, those oils continue to evaporate gradually, releasing aroma into the air. That aroma carries quickly. Smell is the only sense that travels directly to regions of the brain involved in emotion and memory, before passing through higher analytical centers. Signals from the nose move to the olfactory bulb and then into the limbic system, including areas that help encode memory. This is one reason certain scents feel immediate and familiar. The brain does not pause to categorize them first. It responds. Cinnamon often becomes linked to seasonal rituals, to kitchens, to shared meals. The association is not stored in the spice itself, but in neural pathways formed through repetition. Each time scent and experience overlap, connections strengthen. Later, when the scent appears alone, it can activate the network again, a quiet reactivation. Not dramatic, just efficient. So within a small brown curl of dried bark, several systems are at work. Plant tissue responding to moisture loss, aromatic molecules drifting into air, sensory receptors translating chemistry into warmth, neural circuits linking scent to stored experience. Nothing mystical, only structure, signal, and interpretation. And as we begin here, with something as simple as a spice resting on a shelf, there is already more happening than first appears. What rests in a small jar labeled cinnamon once formed part of a living tree, not a fruit, not a seed, not a leaf. Cinnamon is the inner bark of evergreen trees belonging to the genus Cinnamomum. These trees grow in warm, humid climates, especially in regions of Sri Lanka, southern India, and parts of Southeast Asia. They prefer steady rainfall, filtered sunlight, and soil that drains easily. In these conditions, the trees develop smooth grayish bark on the outside and a softer, paler layer just beneath it. It is this inner layer that becomes the spice. The tree itself can reach heights of 10 to 15 meters if left unharvested. Its leaves are oval and glossy, with visible veins running lengthwise from base to tip. When young, the leaves often appear reddish before turning deep green as chlorophyll accumulates. If crushed, the leaves release a faint aroma, though milder than the bark. The tree produces small yellowish flowers and dark purple berries, but these are not the primary focus of cultivation. It is the bark that carries the highest concentration of aromatic oils, particularly cinnamaldehyde. Harvesting cinnamon does not require cutting the entire tree down. Farmers typically coppice the tree, meaning they cut the main stem close to the ground. This encourages multiple new shoots to grow from the base. Within about 2 years, these shoots are thick enough for harvest. The outer bark is scraped away carefully to expose the inner layer. Then, thin strips of that inner bark are peeled off in long sections. Because the tree regenerates, the process can repeat many times over the plant's lifespan. In managed plantations, a single root system can produce harvestable bark for decades. The inner bark contains specialized plant tissues that once helped transport nutrients through the tree. Beneath the rough protective exterior lies a layer called the phloem. The phloem moves sugars produced by the leaves down toward the roots. It is flexible, fibrous, and rich in essential oils. When separated from the outer bark, this layer retains those oils. As it dries, its structure tightens and curls inward. Drying is essential. Freshly peeled bark is pale and soft. As moisture evaporates, chemical changes occur within the plant fibers. Enzymes interact with aromatic compounds. Oxygen exposure slowly deepens the color. The curling happens naturally because the inner and outer surfaces lose moisture at different rates. This differential contraction creates tension that rolls the strip into layered tubes. These are the cinnamon quills often seen whole. Not all cinnamon quills look the same. True cinnamon, often called Ceylon cinnamon, forms thin, delicate layers that resemble rolled parchment. Cassia varieties, which are more common in supermarkets, form thicker, harder curls with a single dense layer. The botanical difference lies in species. Ceylon cinnamon comes primarily from Cinnamomum verum, while cassia types may come from Cinnamomum cassia or related species. Though both are cinnamon, their structure and chemical composition differ slightly. The concentration of cinnamaldehyde varies among species. Cassia tends to have higher levels, which creates a stronger, sharper flavor. It also contains higher levels of coumarin, a naturally occurring compound that, in large amounts, can stress the liver. Ceylon cinnamon contains much less coumarin and has a softer, more layered taste. These differences are chemical, not merely aesthetic. Once dried, cinnamon becomes shelf-stable because its moisture content drops significantly. Microbial growth slows in low-moisture environments. The essential oils act as mild natural preservatives, inhibiting certain bacteria and fungi. This combination of dryness and chemical defense allows cinnamon to remain usable for long periods when stored away from heat and light. So the spice that sprinkles across oatmeal or dissolves into tea began as part of a living transport system inside a tropical tree. Fibers once moving sugars now carry scent. Structures that once supported growth now shape themselves into spirals as they dry. Even in stillness, the bark reflects the processes that formed it, and the quiet chemistry that began inside a tree continues to unfold long after the harvest is complete. Not all cinnamon is the same, even though it often looks similar once ground into powder. The jar on a kitchen shelf may contain one of several related species, each with its own structure and chemistry. The two most common categories are Ceylon cinnamon and Cassia cinnamon. Both come from trees in the Cinnamomum genus, yet their physical form and chemical balance differ in measurable ways. Ceylon cinnamon, sometimes called true cinnamon, comes primarily from Cinnamomum verum. It is native to Sri Lanka and parts of southern India. When dried, its bark forms thin, delicate quills made of multiple soft layers rolled tightly together. If you break a Ceylon quill, it fractures easily. The layers resemble fine parchment stacked inside one another. The color is light brown, almost tan. The texture is brittle, but not hard. Cassia cinnamon, which includes species such as Cinnamomum cassia and Cinnamomum burmannii, is more widely grown in China, Indonesia, and Vietnam. Its bark is thicker and forms a single dense curl rather than many thin layers. The color is deeper, often reddish-brown. When broken, cassia feels firm and woody. It does not crumble in the same delicate way as Ceylon cinnamon. These structural differences arise from the thickness of the inner bark and how it dries. Cassia bark is cut in thicker sections, and its cellular layers contain slightly different proportions of fibrous tissue. As moisture leaves the bark, the tension produces a tighter, denser curl. The variation is botanical rather than cosmetic. The plant species themselves have evolved with distinct growth patterns and chemical compositions. Chemistry explains much of the flavor contrast. Cassia cinnamon contains higher concentrations of cinnamaldehyde, the compound responsible for the sharp, warming sensation described earlier. This higher concentration produces a stronger, more intense taste. It is bold and direct. Ceylon cinnamon contains cinnamaldehyde as well, but in slightly lower amounts, along with additional aromatic compounds that create a softer, more complex profile. Many describe it as subtle and layered. Another compound, coumarin, appears in higher levels in Cassia cinnamon. Coumarin occurs naturally in various plants and contributes to aroma, but in large quantities it can affect liver function. Regulatory agencies in several countries set recommended limits for daily coumarin intake. Because cassia contains more of it, consistent heavy consumption of cassia cinnamon over time may approach those limits. Ceylon cinnamon contains much smaller amounts, often considered negligible by comparison. When cinnamon is ground into powder, visual differences become harder to detect. The layered structure disappears into fine particles. Most commercially available powdered cinnamon in North America and Europe is cassia, partly because cassia trees grow in larger quantities and yield thicker bark, which increases production efficiency. Ceylon cinnamon is more delicate and requires more careful processing. Its global supply is smaller, and its price is often higher. If you examine whole quills side by side, the distinction becomes clear. One appears as a tight scroll of thin sheets. The other looks like a solid curl with a single thick wall. Both release aroma when broken. Both carry warmth. Yet their origin stories differ slightly, shaped by geography, climate, and plant lineage. Climate plays a role in this variation. Sri Lanka's consistent tropical humidity supports the growth of Cinnamomum verum, while other regions favor cassia species adapted to slightly different soil and rainfall patterns. Over centuries, cultivation practices reinforced these distinctions. Farmers selected trees for yield, resilience, and flavor strength. Subtle genetic differences accumulated. In powdered form, these distinctions often blend into routine familiarity. A spoonful stirred into batter reveals little about the tree from which it came. Yet within that powder lies the biological history of a specific species, grown in a particular climate, harvested at a certain thickness, dried under particular conditions. The sensory experience remains warm and recognizable, but beneath that familiarity rests botanical variation shaped by geography and time. Before cinnamon becomes a spiral resting in a glass jar, it passes through a sequence of quiet physical steps. The transformation from living bark to dried quill depends less on machinery and more on timing, moisture, and careful handling. The process begins when young shoots from the cinnamon tree reach the right thickness, usually after about 2 years of growth. At this stage, the bark contains a balanced concentration of aromatic oils and flexible plant fibers. Farmers cut the shoots close to the base. This method encourages new stems to grow from the same root system, allowing the plant to regenerate repeatedly. The outermost bark is then scraped away with curved blades. This outer layer is rough and protective, designed to shield the tree from insects and environmental stress. It contains less of the aromatic compounds that define cinnamon. Removing it exposes the inner bark, which is smoother and lighter in color. The inner bark is carefully loosened and peeled off in long strips. This step requires skill. If the strips tear or crack unevenly, the resulting quills will be shorter or less uniform. The peeled bark at this stage is moist and pale. It feels flexible, almost leathery. Its scent is present, but not yet concentrated. The aromatic oils are still distributed within plant tissues that contain significant water. Drying begins almost immediately. The strips are laid flat or draped over supports in open air, often under shade to prevent excessive heat from degrading volatile compounds. As water slowly evaporates from the bark, its structure begins to change. Plant cells that once carried nutrients lose internal pressure. Fibers tighten. Because the inner and outer surfaces dry at slightly different speeds, tension builds unevenly across the strip. This uneven contraction causes the bark to curl inward from both sides, gradually forming the familiar scroll shape. The curling is not forced. No tool shapes it into spirals. It is a natural consequence of moisture gradients within plant tissue. As drying continues, smaller strips may be inserted into larger ones, forming longer quills composed of multiple layers. In salon cinnamon production, thin pieces are often nested together to create uniform lengths before final drying. Cassia production typically involves thicker single layers that curl on their own without layering. During drying, oxidation also occurs. Oxygen interacts with compounds inside the bark, subtly deepening its color from pale beige to warm brown. Enzymatic reactions modify certain aromatic molecules, refining the scent profile. The intensity of cinnamaldehyde becomes more noticeable as water content decreases. Volatile oils become concentrated within a smaller volume of material. Drying must proceed at a controlled pace. If bark dries too quickly under direct sun or high heat, cracking can occur, and aromatic compounds may dissipate prematurely. If drying is too slow in overly humid conditions, microbial growth becomes possible. The balance between airflow, shade, and time determines final quality. Traditional producers rely on experience rather than digital sensors, observing color, texture, and flexibility to judge readiness. Once fully dried, the quills become firm and brittle. Moisture content drops to a level that discourages mold and bacterial growth. The spice becomes shelf-stable. At this point, cinnamon may be cut into shorter pieces or ground into powder. Grinding dramatically increases surface area, allowing more aromatic molecules to escape at once. This is why freshly ground cinnamon smells stronger than pre-ground spice that has sat for months. The journey from bark to spice is not dramatic. There are no sudden transformations. Instead, slow evaporation reshapes structure. Fiber contracts, color deepens, aroma intensifies. A living transport layer that once moved sugars through a tree becomes a preserved spiral carrying scent across kitchens and markets. The shape that forms during drying reflects the quiet physics of plant tissue responding to air. Even after harvest, the material continues to behave according to simple physical rules. And through those rules, cinnamon acquires the form most people recognize without ever having seen the tree itself. Within each cinnamon quill, the scent and warmth are carried by a small collection of chemical compounds. The most prominent of these is cinnamaldehyde. This molecule makes up a large percentage of cinnamon's essential oil and is responsible for much of its recognizable aroma. Chemically, cinnamaldehyde belongs to a group of organic compounds called aldehydes. Its structure contains a carbon chain with a reactive aldehyde group at one end. That structure allows it to interact readily with receptors in the body and with oxygen in the air. When cinnamon bark is intact, cinnamaldehyde is stored within specialized plant cells. These cells protect the compound until the bark is cut, dried, or ground. Crushing the bark breaks open those cells, releasing volatile molecules into the air. Because cinnamaldehyde is volatile, it evaporates easily at room temperature. This evaporation is what allows the scent to travel from a spoonful of cinnamon to the nose. The nose contains olfactory receptors that bind to specific molecular shapes. Cinnamaldehyde fits into certain receptors in a way that triggers a distinct pattern of electrical signals. Those signals move along the olfactory nerve toward the brain, where they are interpreted as the familiar scent of cinnamon. Each scent corresponds to a unique combination of receptor activations, almost like a pattern of keys being pressed on a piano. Cinnamon's pattern is consistent enough that the brain recognizes it quickly. Cinnamaldehyde also interacts with sensory nerve endings in the mouth and nasal passages. It activates channels involved in detecting chemical irritation and warmth. These channels, part of a family known as transient receptor potential channels, respond to changes in chemical environment. When cinnamaldehyde binds to them, the nervous system interprets the activation as mild heat. Unlike capsaicin, which strongly stimulates pain receptors and can create a burning sensation, cinnamaldehyde produces a softer, more diffuse warmth. The body registers change without sharp discomfort. Cinnamon contains other compounds as well. Eugenol, which is also found in cloves, contributes subtle sweetness and depth. Linalool adds faint floral notes. Coumarin appears in higher concentrations in cassia varieties and has its own mild, sweet aroma. These compounds exist together within the bark's essential oil, creating a layered sensory profile rather than a single sharp scent. The balance of these molecules varies by species, soil conditions, and drying method. Essential oils in cinnamon serve a protective purpose for the tree. Aromatic compounds can deter insects and inhibit certain fungi or bacteria. From a biological perspective, cinnamaldehyde is part of the tree's defense system. Plants cannot move away from threats, so they rely on chemical barriers. The same molecule that creates warmth on the tongue once functioned as a protective signal within living bark. Exposure to air gradually changes cinnamon's chemistry. Oxygen reacts with aldehydes, slowly altering their structure. Heat accelerates this process. This is why ground cinnamon loses potency over time. The increased surface area allows volatile compounds to escape more quickly, and oxidation reduces their concentration. Whole quills retain aroma longer because their inner layers remain protected from air. Breaking a quill releases a new wave of scent as trapped oils are exposed. In laboratory studies, cinnamaldehyde has shown antimicrobial activity against certain bacteria and fungi. The molecule can disrupt microbial cell membranes, interfering with growth. This effect is part of why cinnamon has been used historically in food preservation. Though not strong enough to replace modern preservation methods on its own, the compound contributes mild protective properties. Inside a simple stick of cinnamon, then, lies a network of organic molecules interacting with air, receptors, and time. Chemistry explains the scent drifting upward, the warmth spreading across the tongue, and the gradual fading of aroma on a shelf. Nothing abstract, only molecular structure meeting biological interpretation. The experience of cinnamon begins at this small scale, where atoms arrange into shapes that the nervous system knows how to read. When people describe cinnamon as smelling warm, they are not referring to temperature in the air. The room does not heat. The air does not thicken. Yet the perception shifts. The scent feels round, comforting, almost glowing. That feeling begins in the nose, where odor molecules dissolve into a thin layer of mucus lining the nasal cavity. There, they meet olfactory receptors embedded in specialized sensory neurons. Each olfactory receptor is shaped to respond to certain molecular features. Humans carry hundreds of receptor types, each tuned to slightly different chemical structures. When cinnamaldehyde drifts upward and binds to its matching receptors, those neurons send electrical signals along thin fibers toward the olfactory bulb at the base of the brain. From there, the signal travels in a way that is unique among the senses. Sight, sound, and touch first pass through the thalamus, a relay station that filters and routes information before it reaches higher processing centers. Smell is different. Olfactory signals project directly into parts of the limbic system, including the amygdala and hippocampus. These regions are involved in emotion and memory formation. Because of this direct connection, scent can feel immediate. It reaches emotional circuits without waiting for layered analysis. This is one reason cinnamon's aroma can feel warmer than its chemical properties alone might suggest. The molecule itself activates receptors that contribute to mild warmth perception. But the brain also layers interpretation onto the signal. If past experiences linked cinnamon with baking, gatherings, or seasonal rituals, those associations become part of the sensory response. The scent arrives, and neural networks formed through repetition activate in parallel. The perception expands beyond simple odor into familiarity. Memory linking does not require conscious recall. The hippocampus encodes experiences as patterns of neural activity. When a familiar scent appears, it can partially reactivate those patterns. The result may not be a clear image or story. It may simply be a shift in internal state. Heart rate may slow slightly, muscles may soften, attention may turn inward. The change is subtle yet measurable. Studies show that pleasant familiar scents can reduce activity in brain regions associated with threat monitoring. Cinnamon's aroma contains compounds that are both sweet and slightly sharp. Sweetness in scent often signals ripeness or energy-rich foods. Sharpness can signal potency. Together, they create a balanced profile that the brain interprets as safe but active. The warmth perception comes partly from trigeminal nerve involvement, which detects chemical irritation and contributes to sensations like coolness from menthol or heat from chili. Cinnamaldehyde stimulates these pathways gently, adding to the impression of internal heat without pain. Perception is not a direct copy of the outside world. It is a construction shaped by sensory input and past experience. When cinnamon enters the air, the molecules do not carry meaning on their own. They bind to receptors, trigger signals, and the brain assembles those signals into a feeling. Warmth is not in the molecule. It is in the interpretation of receptor activity, combined with stored patterns. Even the act of inhaling cinnamon alters internal rhythms. Slow breathing draws volatile compounds across receptors. The olfactory system adapts quickly, meaning strong scents fade after continuous exposure. This adaptation prevents overload and allows new odors to be detected. With cinnamon, the initial perception may feel vivid, then soften as receptors reduce firing rate. The shift itself can feel calming, as intensity gives way to gentler awareness. So when cinnamon smells warm, what is happening is layered. A volatile molecule meets a receptor. Electrical signals move directly into emotional centers. Stored associations activate. The trigeminal system adds a mild thermal impression. perception shifts slightly toward comfort and internal heat, even though the surrounding air remains unchanged. The warmth is not in the spice alone. It is in the way the nervous system translates chemistry into experience. The nervous system is always adjusting. It measures light, sound, movement, scent. It decides, often without conscious thought, whether the environment requires alertness or allows ease. Cinnamon enters this system quietly. Its aroma does not demand attention in the way a sudden noise does. It does not signal danger. Instead, it produces a mild shift in internal state, one that combines gentle stimulation with familiarity. When cinnamaldehyde and related aromatic compounds activate receptors in the nose, signals travel toward the limbic system and other connected regions. These areas help regulate emotional tone and autonomic responses. The autonomic nervous system has two primary branches. The sympathetic branch prepares the body for action. The parasympathetic branch supports rest and digestion. Most sensory input contains some element of evaluation. Is this new? Is it threatening? Is it safe? Familiar scents tend to reduce vigilance. Research shows that when an odor is recognized and associated with neutral or positive experiences, activity in brain regions tied to threat detection decreases slightly. The amygdala, which helps evaluate potential danger, responds differently to known pleasant smells than to unfamiliar or unpleasant ones. Cinnamon, when linked with routine and repetition, often falls into the category of safe familiarity. This familiarity does not eliminate mild stimulation. Cinnamaldehyde still activates sensory channels. The trigeminal nerve contributes a subtle tingling warmth. The brain registers the chemical presence. Yet because the pattern is predictable, the overall nervous response may remain balanced. Mild activation without alarm. Signal without urgency. Warm spices in general share this quality. Unlike sharp synthetic scents that can overwhelm receptors, natural aromatic compounds tend to disperse gradually. Cinnamon's volatile oils evaporate steadily, not explosively. As concentration rises and falls, receptors adapt. Adaptation is a protective mechanism. After a short period, the firing rate of olfactory neurons decreases even if the scent remains present. This fading reduces overstimulation. The nervous system conserves energy by filtering constant input. In this way, cinnamon can feel calming, even though it produces mild stimulation at the receptor level. The stimulation is brief and predictable. The familiarity signals safety. The balance between these two effects can shift internal tone toward steadiness. Heart rate variability studies suggest that pleasant odors may increase parasympathetic activity, supporting relaxation responses. While cinnamon has not been studied as extensively as lavender in this context, the broader pattern of scent familiarity reducing vigilance applies. There is also a learning component. Repeated exposure to a scent in calm environments strengthens associative networks. If cinnamon is often encountered during quiet routines, such as drinking tea or preparing food at home, the nervous system encodes that pairing. Later, when the scent appears again, it can cue the same physiological patterns. This is classical conditioning at a subtle level. The body anticipates what usually follows. If calm typically follows cinnamon, calm becomes part of the response. The nervous system prefers predictability. Sudden changes increase alertness. Stable patterns decrease it. Cinnamon's aroma, once recognized, becomes part of a stable pattern. Its molecular structure does not change from one encounter to the next. Receptors fire in similar sequences. Neural circuits recognize the pattern and categorize it quickly. Recognition requires less processing effort than novelty. Reduced processing effort can translate into reduced tension. Even breathing patterns may shift slightly. Pleasant, familiar scents can encourage slower inhalation. Slow breathing influences vagal tone, a measure of parasympathetic engagement. Though the effect is modest, small shifts accumulate. The nervous system continuously integrates sensory input with internal regulation. Cinnamon contributes to that integration through chemistry and repetition, rather than force. So within the quiet presence of a spice, multiple systems cooperate. Sensory receptors activate. Emotional circuits assess familiarity. Autonomic balance adjusts by small degrees. The result is not sedation. It is not strong stimulation. It is a mild internal warming paired with a sense of known pattern. In that balance, the nervous system often chooses steadiness over alertness, ease over vigilance. Beyond its scent and warmth, cinnamon has drawn attention for its possible effects inside the body, particularly in how it relates to blood sugar regulation. Blood sugar, or glucose, is the primary fuel circulating in the bloodstream. After eating carbohydrates, glucose levels rise. In response, the pancreas releases insulin, a hormone that helps cells absorb glucose and use it for energy. This process keeps blood sugar within a stable range. Researchers began investigating cinnamon's role in this system several decades ago. Laboratory studies found that certain compounds in cinnamon, including cinnamaldehyde and related polyphenols, appeared to influence how cells respond to insulin. Some experiments suggested that cinnamon extracts might enhance insulin sensitivity, meaning cells could respond more effectively to the hormone. In controlled settings, this sometimes led to modest reductions in fasting blood glucose levels. One proposed mechanism involves insulin receptor activity. Insulin binds to receptors on cell surfaces, triggering a cascade of signals that allow glucose transporters to move to the cell membrane. These transporters, especially one called GLUT4 in muscle and fat tissue, help shuttle glucose from the bloodstream into cells. Some laboratory findings suggest that cinnamon compounds may influence this signaling cascade, potentially supporting glucose uptake. Another area of study focuses on cinnamon's antioxidant properties. Cinnamon contains polyphenols, plant compounds known to reduce oxidative stress in laboratory models. Oxidative stress can interfere with insulin signaling pathways. By reducing this stress, polyphenols may indirectly support metabolic balance. However, much of this evidence comes from cell cultures or animal studies rather than long-term human trials. Human studies have produced mixed results. Some clinical trials report small decreases in fasting blood glucose or improvements in certain metabolic markers when participants consume cinnamon regularly. Other studies show little to no measurable effect. Differences in cinnamon species, dosage, participant health status, and study duration make conclusions complex. Cassia and Ceylon cinnamon contain different levels of active compounds, which may influence outcomes. It is also important to consider scale. The amounts of cinnamon used in some research settings are higher than what most people would use casually in cooking. While culinary use is generally safe for most individuals, concentrated extracts or high daily intake of cassia cinnamon may introduce higher levels of coumarin. As mentioned earlier, coumarin in large amounts can affect liver function. This is why health authorities often recommend moderation rather than heavy supplementation without guidance. Cinnamon does not replace medical treatment for diabetes or metabolic conditions. Blood sugar regulation is influenced by many factors: diet, activity level, genetics, sleep patterns, and hormonal balance. Cinnamon, if it plays a role at all, appears to act as a small contributor within a larger system. The evidence suggests potential, not certainty. What makes this research interesting is not the promise of dramatic change, but the reminder that plant compounds interact with human physiology in measurable ways. A molecule that once served as chemical defense within bark can interact with enzymes, receptors, and transport proteins in the body. The interaction is subtle. It does not override the body's own regulatory systems. Instead, it may adjust signaling pathways slightly, like a gentle influence, rather than a forceful intervention. Metabolism itself is a continuous balancing act. Glucose rises and falls throughout the day. Insulin pulses in response. Cells open and close channels as needed. Cinnamon's compounds enter this dynamic environment and participate briefly before being metabolized by the liver and excreted. Their presence is temporary. When cinnamon is stirred into tea or oatmeal, its impact on blood sugar, if present, is modest and context-dependent. The science continues to explore these mechanisms with caution. What remains consistent is that cinnamon's influence operates at the molecular level, interacting with receptors and enzymes in ways that reflect its chemical structure. The same aldehyde that signals warmth in the mouth can also engage pathways involved in metabolism. And within the body's steady rhythms, these small interactions unfold quietly, without spectacle, as part of a larger physiological conversation. Well before refrigeration, people relied on salt, drying, smoke, and certain plants to slow the spoilage of food. Cinnamon became part of that quiet effort, not because it could stop decay entirely, but because its chemical structure offered mild protection against certain microorganisms. The same compounds that give cinnamon its scent also interact with bacterial and fungal cells in measurable ways. Cinnamaldehyde, the primary aromatic compound in cinnamon, has been shown in laboratory settings to inhibit the growth of some bacteria and fungi. It can disrupt microbial cell membranes, altering permeability and interfering with essential processes inside the cell. When the integrity of a membrane is compromised, the organism struggles to maintain balance. In controlled experiments, this effect reduces growth rates of specific strains. Cinnamon also contains eugenol and other phenolic compounds, which contribute to antimicrobial activity. These molecules can interfere with enzyme systems inside microbes or affect their structural stability. The concentrations required for strong antimicrobial effects in laboratory environments are often higher than those used casually in cooking. Still, even modest levels may contribute to slowing spoilage when combined with other preservation methods. In historical kitchens, spices were often paired with salting or drying techniques. A piece of meat preserved with salt and rubbed with aromatic spices would have reduced moisture and chemical deterrents present simultaneously. Lower moisture content limits bacterial growth because microbes require water to multiply. Cinnamon itself, once dried, contains very little moisture. Its low water activity prevents it from supporting microbial growth within the spice. When added to foods, it contributes aromatic compounds without introducing additional moisture. Temperature also influences preservation. In cooler climates, winter air acted as a natural refrigerator. In warmer regions, drying under sun and airflow was essential. Cinnamon, imported along trade routes, was often used in dishes where long cooking times further reduced microbial risk. Stews, sauces, and preserved fruits sometimes included cinnamon not only for flavor, but also for its subtle protective qualities. The idea that spices were used solely to mask spoiled meat is a common myth. Historical records show that spices were expensive and valued. They were unlikely to be wasted on food that was already unsafe. Instead, their antimicrobial properties may have complemented other preservation practices. Cinnamon's role was supportive rather than dominant. Modern research continues to explore cinnamon oil as a natural preservative. In food science studies, cinnamon essential oil has demonstrated inhibitory effects against certain pathogens, including some strains of Escherichia coli and Staphylococcus aureus, in controlled environments. These findings highlight the biological activity of its components. However, using concentrated essential oils directly in food requires careful regulation because high doses can alter taste and safety. Preservation is ultimately about slowing change. Microorganisms break down tissues, producing acids, gases, and off flavors. Cinnamon's compounds can disrupt parts of this process, extending freshness slightly under the right conditions. The effect is incremental. It does not sterilize food. It adjusts the rate of microbial growth. Even today, cinnamon appears in preserved fruit compotes, pickles, and syrups. Its aromatic oils blend with sugar and acidity to create environments less favorable to spoilage. Sugar reduces water availability. Acidity lowers pH. Cinnamon contributes chemical deterrence. Together, these factors form a layered system of protection. The science behind this preservation lies in molecular interaction. A phenolic compound encounters a bacterial membrane. The membrane structure weakens. Growth slows. Over time, this translates into extended shelf life under appropriate storage. It is chemistry at the microscopic level influencing everyday food practices. So cinnamon's presence in historic kitchens was not only about warmth of flavor. It was part of a broader understanding that certain plants helped maintain food stability. The same bark that curled and dried under tropical air carried compounds capable of influencing microbial survival. Even in stillness, its molecules remained active, participating in quiet chemical exchanges long after harvest. Long before cinnamon rested in glass jars or drifted through modern kitchens, it traveled across deserts and seas toward one of the earliest great civilizations. In ancient Egypt, cinnamon was not a common household spice. It was rare, imported, and associated with ceremony. The dry climate along the Nile preserved many organic materials, and written records along with residue analysis from burial sites, suggest that aromatic resins and spices, including forms of cinnamon or related cassia, were used in ritual contexts. Egyptian embalmers worked with careful attention to preservation. The body was treated with salts, oils, and aromatic substances to slow decay. While myrrh and frankincense were primary components, cassia and cinnamon-like materials were also part of certain mixtures. These aromatic compounds contributed not only fragrance, but also mild antimicrobial properties. The use of such substances aligned with the broader goal of delaying decomposition. In warm climates, preservation required layered approaches. Natron, a naturally occurring salt mixture, removed moisture from tissues. Resins sealed surfaces. Aromatic plant compounds, including those from cinnamon relatives, added scent and possibly additional chemical deterrents. Cinnamon's cinnamaldehyde and related compounds, as discussed earlier, have inhibitory effects on certain microorganisms. In this setting, scent and chemistry work together. Cinnamon did not grow in Egypt. It arrived through long-distance trade networks that connected the Nile Valley with regions of the Arabian Peninsula, East Africa, and South Asia. Traders transported spices in caravans or by sea along routes that predated the later Silk Road. Because the true origin of cinnamon trees was often concealed, the spice carried an aura of mystery. Its distant source increased its perceived value. Ancient texts sometimes describe cinnamon in poetic terms, emphasizing its rarity and fragrance. It was used in temple offerings, perfumed oils, and ceremonial incense. In religious contexts, scent played a symbolic role. Rising smoke carried aroma upward, reinforcing ideas of transcendence and communication with the divine. Cinnamon's volatility made it suitable for such rituals. When heated or burned, its essential oils dispersed quickly into the air, filling enclosed spaces with warmth. At certain points in history, cinnamon's value relative to weight rivaled that of precious metals. Though exact economic comparisons vary, records suggest it was treated as a luxury commodity reserved for elites. Its scarcity came not only from limited supply, but from controlled distribution. Traders sometimes protected their roots by spreading myths about dangerous lands where cinnamon supposedly grew, discouraging direct sourcing. The association with burial rites added symbolic weight. In a culture deeply invested in the concept of afterlife continuity, preservation of the body was essential. Materials used in mummification were chosen for both practical and spiritual reasons. Cinnamon's fragrance may have masked odors during preparation, but it also signaled care and status. Aromatic compounds lingered in tomb chambers, merging scent with ritual memory. Chemical analysis of residues from ancient containers has identified compounds consistent with cinnamon relatives in certain archaeological contexts. While precise identification can be complex due to degradation over millennia, evidence supports the presence of cassia-type spices in embalming mixtures and perfumed oils. These findings illustrate how plant chemistry intersected with early preservation science. Trade routes that carried cinnamon into Egypt laid groundwork for broader exchange networks. Spices moved alongside gold, textiles, and precious stones. With each shipment, knowledge of distant regions expanded. Cinnamon became part of a system that connected climates, languages, and technologies long before global maps were drawn. In the still air of a burial chamber, sealed against the desert, aromatic molecules from imported bark once drifted upward during rituals. The same volatile compounds that now rise from a cup of tea once mixed with resins and oils under torchlight. In both cases, chemistry remained constant. Cinnamaldehyde evaporated, receptors detected scent, and perception followed. Across centuries, the molecule did not change. Only its setting did. As trade networks expanded beyond the Nile and across the Mediterranean, cinnamon continued its quiet movement from tropical forests into the centers of power. By the time of the Roman Empire, spices had become symbols of wealth and reach. Rome did not grow cinnamon. The climate was wrong. Yet through maritime routes and overland caravans, the bark found its way into Roman markets, carried in small bundles that had traveled thousands of miles. Roman merchants relied on established trade pathways that connected the Mediterranean to the Red Sea and beyond. Ships sailed from Egyptian ports such as Berenice and Myos Hormos down the Red Sea, then eastward toward Arabian and Indian harbors. From there, goods moved through complex exchange systems involving multiple intermediaries. Cinnamon likely passed through many hands before reaching Rome. Each exchange increased its price. The Romans valued cinnamon for its aroma and rarity. It was used in perfumes, incense, and certain dishes among the wealthy. In culinary contexts, spices were often blended into sauces combining sweet, sour, and savory elements. Roman taste favored contrast. Cinnamon's warmth complemented honey, wine reductions, and fermented fish sauces such as garum. The spice became part of elaborate banquets that displayed status rather than necessity. At the same time, cinnamon maintained ritual significance. Incense mixtures burned during ceremonies often included aromatic resins and imported spices. When heated, cinnamon released volatile oils quickly, filling enclosed spaces with scent. Smoke rising from altars carried fragrance upward, reinforcing the sensory experience of worship. The volatility of cinnamaldehyde meant that even a small amount could scent a large area. The true origin of cinnamon trees remained obscured. Traders protected supply chains by limiting information. Some ancient writers describe cinnamon as growing in distant lands, guarded by mythical creatures or harvested from inaccessible cliffs. These stories were not accidental. Secrecy preserved monopoly. If buyers believed the source was remote and dangerous, they were less likely to attempt direct procurement. This concealment reflects a broader economic principle. Control of information increases control of value. Cinnamon's physical form, a dried bark curl, gave little indication of its botanical origin. Without knowledge of the tree or region, consumers depended on merchants. The bark itself traveled silently, offering no visible map of its homeland. Roman records show that spices could command extraordinary prices relative to everyday goods. Although exact comparisons vary across sources, cinnamon's cost made it inaccessible to most citizens. Its presence in a household signaled wealth. In this way, plant chemistry intersected with social hierarchy. The same cinnamaldehyde molecules that stimulated receptors in the nose also signaled economic status when they drifted from a banquet hall. Trade routes that delivered cinnamon to Rome were precursors to later Silk Road exchanges. While the classic Silk Road connected East Asia to Europe through Central Asia, maritime spice routes were equally influential. Seasonal monsoon winds allowed ships to cross the Indian Ocean more efficiently, shortening travel times. Knowledge of these wind patterns improved reliability, increasing spice flow. Each stage of the journey exposed cinnamon to new climates. Stored in dry conditions, the bark's low moisture content protected it from spoilage. Essential oils acted as mild deterrents to microbial growth during transit. This stability made cinnamon well suited to long-distance trade. Unlike fresh produce, it did not decay quickly. Its chemistry supported its economics. So in Roman markets, small coils of bark represented far more than flavor. They embodied maritime navigation, guarded knowledge, and shifting winds across oceans. They reflected the capacity of dried plant tissue to endure travel without losing scent. And within each quill, volatile molecules waited. When broken or heated, they released the same warmth sensed centuries earlier in distant forests. Across empires and trade networks, the molecule remained constant. Only the roots surrounding it expanded. As the Roman Empire receded and Europe entered the medieval period, cinnamon did not disappear. Instead, its path shifted. Trade routes grew more complex, and control over spices became a driving force behind maritime exploration. Cinnamon, small and lightweight, traveled farther than most goods. It could cross oceans without spoiling. It retained value in every port. And because it grew only in specific tropical regions, demand in Europe depended entirely on long-distance shipping. By the Middle Ages, Sri Lanka had become widely recognized as a primary source of true cinnamon. The island's humid climate and steady rainfall supported the growth of Cinnamomum verum. Cinnamon trees flourished there, and skilled harvesters developed methods of peeling and drying bark with precision. The spice left Sri Lanka by ship, moving through Indian Ocean trade networks toward the Arabian Peninsula and onward into Mediterranean markets. For centuries, much of this trade was controlled by Arab merchants, who maintained strategic access to maritime routes. They carried cinnamon and other spices northward into ports like Alexandria, where Venetian traders purchased them for distribution across Europe. Each intermediary increased the price. The journey from tree to European table involved layers of negotiation, risk, and distance. Because cinnamon was compact and high in value, it played a disproportionate role in shaping global shipping routes. European powers sought direct access to spice-producing regions in order to bypass middlemen. The search for shorter, controlled sea routes intensified in the 15th century. Portuguese explorers sailed around the southern tip of Africa, reaching Indian Ocean waters and eventually establishing a foothold in Sri Lanka in the early 16th century. Control over cinnamon groves became a strategic priority. Colonial competition followed. The Portuguese, then the Dutch, and later the British, sought authority over Sri Lanka's cinnamon trade. Fortifications were built. Harvesting was regulated. Cinnamon exports became part of imperial economic systems. Though the spice itself remained physically unchanged, a dried curl of bark, the political and economic structures surrounding it transformed dramatically. Shipping routes that once depended on seasonal monsoon winds became increasingly formalized. European navies protected merchant vessels. Ports were fortified. Cinnamon was loaded into wooden crates and stored in dry holes to preserve its aroma during long voyages. Its low moisture content once again supported its stability. Unlike fresh crops, it could endure months at sea without significant degradation, provided it remained dry. Cinnamon's influence extended beyond flavor. The pursuit of spices helped accelerate navigational advancements, map-making, and ship design. Mariners studied wind patterns, ocean currents, and celestial navigation in order to secure reliable routes. Though spices were only part of broader trade motivations, cinnamon contributed to the economic justification for exploration. The result was a reconfiguration of global exchange. Maritime exploration connected continents more directly than ever before. Cinnamon, harvested from smooth-barked trees in humid forests, now shaped decisions made in European courts. Control of its trade signaled power. In the quiet curl of a cinnamon quill rests a record of these movements. It traveled from tropical grove to colonial port, from guarded harbor to distant market stall. Each shipment represented coordination across oceans, yet at its core, the spice remained plant tissue dried under shade, carrying volatile oils that would release warmth when broken. The roots changed. Empires rose and shifted, but the chemistry inside the bark did not alter. Cinnamaldehyde evaporated into the air in medieval kitchens, just as it does today. The same molecule that once scented royal banquets now rises from simple cups of tea. And behind that scent lies a history of ships, winds, and contested coastlines shaped in part by the demand for cinnamon. Cinnamon does not begin as powder or even as a curled quill. It begins as an evergreen tree rooted in warm soil, drawing water upward through slender trunks and glossy leaves. Most commercial cinnamon comes from species within the Cinnamomum genus, particularly Cinnamomum verum and related cassia species. These trees belong to the laurel family, the same broader plant family that includes bay laurel. In their natural environment, cinnamon trees grow in tropical climates where rainfall is steady and temperatures remain warm year-round. Sri Lanka, southern India, parts of Indonesia, Vietnam, and southern China provide the right balance of humidity, filtered sunlight, and well-drained soil. The air in these regions often carries moisture, and the ground rarely freezes. Under these conditions, cinnamon trees can grow steadily without seasonal dormancy. A mature cinnamon tree may reach 10 to 15 meters in height if left unpruned. Its trunk is relatively slender compared to larger forest trees. The bark on the outside appears grayish-brown and somewhat rough. Beneath this outer layer lies the smoother inner bark that becomes the spice. The leaves are oval with smooth edges and a slightly pointed tip. When young, they often emerge reddish or bronze before deepening into glossy green. Veins run parallel from the base toward the tip, creating a subtle pattern visible when light passes across the surface. If you crush a fresh cinnamon leaf between your fingers, it releases a faint aroma, though less concentrated than the bark. The essential oils are distributed throughout the plant, but the inner bark contains the highest concentration of cinnamaldehyde. The tree also produces small clusters of pale yellow flowers, followed by dark purple berries. These berries are not commonly harvested for spice production, but they mark the reproductive cycle of the tree. Cinnamon cultivation often relies on a method called coppicing. Instead of allowing a single tall trunk to dominate, Farmers cut the tree back close to ground level after a few years of growth. This encourages multiple new shoots to emerge from the root base. These shoots grow quickly in humid conditions, forming straight stems ideal for bark harvesting. After about 2 years, the stems are thick enough to peel. The cycle can repeat for decades because the root system remains intact. The tree's ability to regrow bark after harvesting is central to sustainable production. When the inner bark is carefully removed from young shoots, the remaining root network continues to supply nutrients to new growth. Unlike trees harvested for timber, cinnamon trees are not typically uprooted or fully destroyed. Instead, they regenerate repeatedly. This regrowth reflects the resilience of tropical evergreen species, adapted to recover from natural damage such as storms or grazing. Inside the bark, vascular tissues once carried sugars produced by photosynthesis from leaves to roots. The phloem, flexible and fibrous, contains the aromatic compounds that later define cinnamon's scent. When the bark is peeled and dried, these tissues lose moisture, but retain their chemical composition. The tree itself, still rooted in warm soil, continues drawing water upward through xylem channels, producing new shoots for future harvests. In the forest, cinnamon trees grow among other tropical plants, competing for light and nutrients. Their evergreen leaves allow photosynthesis throughout the year. The glossy surface of each leaf reflects some sunlight, reducing water loss while still absorbing energy. This steady process supports the growth of bark layers rich in aromatic oils. Behind every small curl of dried spice stands a living tree shaped by humidity, soil, and careful pruning. Smooth leaves gather sunlight. Roots draw water upward. Bark layers thicken quietly over time. And from this continuous cycle of growth and renewal, the inner layer emerges, ready to curl under open air, carrying within it the chemistry of the forest. If you hold a cinnamon stick between your fingers, the first sensation is texture. It feels dry, firm, and slightly rough along the outer surface. The inner layers, visible where the quill curls inward, appear smooth and thin, almost like rolled parchment. This physical structure is the result of drying plant tissue that once carried moisture and nutrients. When water leaves those tissues, flexibility decreases. What remains is a brittle, lightweight form. Brittleness is a property of materials that fracture rather than bend when force is applied. In fresh bark, plant cells are supported by water pressure inside their walls. This internal pressure, known as turgor, keeps tissues flexible. Once harvested and dried, cinnamon bark loses that internal support. The cellulose fibers and lignin structures remain, but without moisture, they cannot flex easily. Instead, they snap. When you break a cinnamon stick, it fractures cleanly, revealing fibrous strands inside. The difference between Ceylon and Cassia cinnamon is noticeable in texture as well. Ceylon quills are composed of many thin layers rolled together. They feel lighter and more delicate. Cassia sticks are thicker, denser, and harder to break. The structural thickness of the bark determines how much resistance it offers when pressure is applied. These physical differences reflect botanical variation rather than processing style alone. Grinding transforms texture completely. When cinnamon is reduced to powder, its surface area increases dramatically. A stick has relatively little exposed area compared to the volume of material inside it. Once ground, thousands of tiny particles are created. Each particle exposes new surfaces to air. This increased surface area allows volatile aromatic compounds to escape more quickly. That is why freshly ground cinnamon smells stronger than a whole stick left intact. Surface area plays a central role in aroma release. Volatile molecules such as cinnamaldehyde evaporate from exposed surfaces. The more surface available, the more molecules can escape at once. Grinding accelerates this process. At the same time, grinding also accelerates oxidation. Oxygen in the air reacts gradually with exposed compounds, altering their structure and reducing potency over time. This is why pre-ground cinnamon loses intensity more quickly than whole quills. With powder, every particle is exposed to air. Oxidative reactions proceed across a wide area. In contrast, whole sticks protect their inner layers. When you snap a quill, you release scent from layers that were previously shielded. The fragrance rises suddenly because those oils have been preserved inside a tight curl. Texture also affects how cinnamon behaves in cooking. A whole stick releases flavor slowly as heat draws essential oils outward into liquid. Powder disperses rapidly, mixing throughout a dish and delivering immediate aroma. The physical form determines extraction rate. In simmering stews or teas, a stick can steep gently, releasing compounds over time. Powder dissolves and spreads quickly, though some particles may settle. Under magnification, cinnamon powder appears as irregular fragments of plant tissue. Fibrous strands, broken cell walls, and tiny splinters compose the fine brown dust. Each fragment contains remnants of vascular tissue that once transported sugars inside the tree. The brittle nature of dried bark allows it to be reduced easily with grinding stones or mechanical mills. Even the sound of cinnamon reflects its texture. A stick tapped lightly against a surface produces a crisp, dry note. It lacks the soft thud of moist wood. That dryness is the result of evaporation completed long ago during curing. The bark that once flexed slightly under tropical humidity now fractures under minimal pressure. So the texture of cinnamon is a record of its transformation. Moist plant tissue became rigid through loss of water. Layered bark curled inward and hardened. Grinding multiplied exposed surfaces and accelerated aroma release. The way cinnamon feels between fingers or breaks under pressure reflects simple physical principles shaped by drying, fiber structure, and time. Within that brittle curl, the forest chemistry waits quietly until surface meets air once again. As cinnamon traveled along maritime and overland trade routes, it settled into kitchens shaped by climate, agriculture, and tradition. Its path across continents left subtle marks on regional cuisines. Where trade networks reached reliably, cinnamon appeared more frequently. Where routes were distant or unstable, its presence was rare and reserved for special occasions. The geography of spice use often mirrors the geography of exchange. In Mexico, cinnamon became woven into drinks and desserts. Mexican hot chocolate traditionally includes cinnamon sticks simmered in milk with cacao and sugar. The spice softens the bitterness of cacao and adds aromatic warmth without sharp heat. In some regions, cinnamon is ground with sugar and sprinkled over pastries or stirred into café de olla, a coffee brewed with cinnamon and piloncillo. These uses reflect centuries of trade between Asia, Europe, and the Americas, especially during the Spanish colonial period when spices move through transoceanic routes linking Manila, Acapulco, and European ports. In the Middle East, cinnamon appears in savory dishes as often as in sweets. It is blended into spice mixtures used for stews, rice dishes, and meat preparations. The balance of sweet and savory flavors is a hallmark of many regional cuisines. Cinnamon contributes warmth without overpowering other ingredients. In some dishes, it is paired with cumin, coriander, and cloves. The spice integrates into slow-cooked meals, where heat draws out its essential oils gradually. Scandinavian baking offers another example. Cinnamon rolls, known in Sweden as kanelbullar, are layered with butter, sugar, and cinnamon before being baked into soft spirals. The scent released during baking fills enclosed spaces quickly, reinforcing seasonal traditions. Northern climates historically relied on imported spices for flavor diversity during long winters. Cinnamon's arrival through trade networks brought a sense of novelty that later became routine. Across North Africa, cinnamon blends into tagines and spiced teas. In Morocco, it can appear in both sweet pastries and savory meat dishes, sometimes dusted lightly over fruit or incorporated into complex spice blends. The spice's flexibility allows it to bridge flavors rather than dominate them. Its mild sweetness complements dried fruits, while its warmth balances richer ingredients. In parts of South Asia, cinnamon bark is added whole to rice dishes and curries. It releases aroma slowly during cooking and is often removed before serving. The bark may be paired with cardamom, cloves, and bay leaves in layered spice mixtures. Because cinnamon trees grow in parts of India and Sri Lanka, access historically differed from regions that relied solely on imports. Proximity influenced frequency of use. Patterns of cinnamon's presence correlate with historical trade routes. Ports connected to Indian Ocean shipping lanes saw earlier and more consistent access. Mediterranean cities integrated spices through established merchant networks. Northern European regions encountered cinnamon through intermediaries and used it sparingly at first. Over time, as shipping became more regular and global commerce expanded, the spice moved from elite tables into everyday kitchens. Cultural memory shapes how cinnamon is perceived in each region. In some places, it signals celebration. In others, it blends quietly into daily meals. The chemistry remains constant, cinnamaldehyde evaporating into warm air, but its social meaning shifts. The same molecule accompanies hot chocolate in Mexico, lamb stew in Lebanon, and sweet buns in Sweden. Trade not only distributed cinnamon geographically, but also influenced how it was combined with local ingredients. Cacao met cinnamon through colonial exchange. Sugar plantations increased the pairing of sweetness and spice. Rice cultivation in Asia provided a neutral base for aromatic bark. Each combination reflects layers of agricultural history and movement. Cinnamon's presence across cultures is not random. It follows shipping lanes, caravan paths, and imperial connections. Its versatility allowed it to adapt to local tastes while maintaining its recognizable warmth. Within kitchens separated by oceans and climates, the brittle bark dissolved into liquids and doughs, releasing aroma that bridged distance through shared chemistry. When cinnamon feels warm, the warmth does not come from temperature. If you place a thermometer in cinnamon powder, it reads the same as the surrounding air. Yet when cinnamon touches the tongue or drifts through the nose, The sensation resembles gentle heat. This effect comes from how certain sensory receptors respond to chemical signals rather than to actual thermal change. The body detects temperature through specialized nerve endings embedded in the skin and mucous membranes. These receptors belong to a family known as transient receptor potential channels, often abbreviated as TRP channels. Different members of this family respond to cold, heat, pressure, or chemical irritants. Menthol activates receptors associated with coolness. Capsaicin, the compound in chili peppers, activates receptors associated with high heat and pain. Cinnamon works through a different pathway. Cinnamaldehyde primarily activates a receptor known as TRPA1. This receptor is sensitive to chemical irritants and environmental signals. When cinnamaldehyde binds to TRPA1 on sensory neurons in the mouth or nasal passages, it triggers an electrical signal. The brain interprets this activation as a warming or tingling sensation. However, the tissue temperature has not changed. No external heat has been added. Capsaicin, by contrast, activates TRPV1 receptors, which are tuned to detect higher temperatures and painful heat. That is why chili peppers can produce intense burning sensations. Cinnamon's activation of TRPA1 is milder and more diffuse. It creates a sense of warmth without the sharp discomfort associated with spicy peppers. The difference lies in receptor type and signal intensity, not in actual temperature. The trigeminal nerve carries these signals to the brain. This nerve monitors chemical irritation, pressure, and temperature within the face and oral cavity. When cinnamaldehyde stimulates TRPA1 receptors, the trigeminal nerve transmits the information to brain regions that integrate sensory input. The result is a perception of internal warmth, layered over the scent detected by the olfactory system. This dual sensory pathway, olfactory and trigeminal, explains why cinnamon feels richer than scent alone. The olfactory receptors identify the aroma. The trigeminal system adds a mild physical sensation. Together, they create a multi-dimensional experience. Even small amounts of cinnamon can engage both pathways simultaneously. The warmth produced by cinnamaldehyde is sometimes described as a sensory illusion. The brain constructs the experience of heat based on receptor activation patterns rather than direct measurement of temperature. Perception depends on interpretation. If TRPA1 channels fire in a pattern associated with warming stimuli, the brain categorizes the input accordingly. The tissue itself remains at baseline temperature. This mechanism highlights a broader principle of sensory biology. The nervous system does not experience the external world directly. It receives electrical signals from receptors tuned to specific chemical or physical changes. Those signals are processed and interpreted. Cinnamon's molecules fit into receptor sites in a way that resembles other warming stimuli. The brain responds based on that resemblance. Even adaptation plays a role. With repeated exposure, TRPA1 receptors may reduce firing intensity, and the warmth sensation can fade. This adaptation prevents continuous overstimulation. It allows the nervous system to recalibrate and remain responsive to new stimuli. When cinnamon is tasted again after a pause, the warmth may return briefly before softening once more. So when cinnamon seems to warm the mouth or chest, the warmth is not thermal energy moving through tissue. It is chemical interaction translated into perception. Cinnamaldehyde binds to TRPA1 receptors. The trigeminal nerve carries the signal. The brain interprets it as heat. Within that interpretation lies the sensation of comfort often associated with warm spices. The temperature remains unchanged. The experience shifts nonetheless. Cinnamon grows in tropical climates. Its trees prefer steady humidity and warm air. There is no frost in the groves where bark is harvested. And yet, in many parts of the world, cinnamon is closely associated with winter. The connection is not botanical. It is cultural and neurological. In colder regions, imported spices once arrived in limited quantities and were often reserved for special occasions. Winter holidays became moments when rare ingredients were used more freely. Cinnamon entered festive breads, cakes, and drinks prepared during colder months. Over time, repetition strengthened this pairing. The scent of cinnamon began to coincide with specific seasonal rituals. Seasonal pairing reinforces memory. When a scent appears consistently alongside particular events, the brain encodes the combination as a pattern. Cinnamon might be present during gatherings, baking sessions, or celebrations that occur at roughly the same time each year. Because these experiences are emotionally meaningful, they leave strong neural traces. Later, when cinnamon's aroma appears alone, it can reactivate parts of those networks. The hippocampus, involved in memory consolidation, works closely with the olfactory system. As mentioned earlier, smell signals travel directly to limbic regions before passing through higher processing centers. This direct pathway allows scent to become tightly linked with emotional experiences. Research suggests that scent memory encoding can be especially strong during childhood. Early experiences with specific smells often remain vivid decades later. If cinnamon was present in early seasonal traditions, those early pairings may shape lifelong associations. A child who smells cinnamon during winter baking may encode the scent alongside feelings of warmth, family presence, or indoor shelter from cold weather. Even if later experiences vary, the early network remains. When the scent returns, the memory pathway activates automatically. Winter itself heightens certain sensory contrasts. Cold outdoor air makes indoor warmth more noticeable. Hot drinks release steam carrying volatile compounds upward toward the nose. Cinnamon, added to warm beverages, disperses readily in rising vapor. The physical warmth of the drink combines with the chemical warmth perceived through TRPA1 receptors. The two sensations align, reinforcing the association between cinnamon and heat. Cultural reinforcement deepens this link. Media, marketing, and shared traditions often highlight cinnamon in seasonal recipes. Holiday foods feature cinnamon prominently. Over decades, repeated exposure strengthens the seasonal script. Even individuals in warmer climates may adopt these patterns through global cultural exchange. Interestingly, cinnamon is not inherently seasonal in tropical regions where it grows. There, it may be used throughout the year. The winter association emerges primarily in climates with strong seasonal contrast. Scarcity of fresh produce during colder months historically increased reliance on preserved foods and spices. Cinnamon's stability and long shelf life made it practical for storage. Its use during winter months became both functional and symbolic. The power of scent memory encoding means that these associations can feel immediate and involuntary. A faint trace of cinnamon may evoke imagery of winter landscapes, even when the current environment is warm. The response arises not from the spice itself, but from neural circuits shaped through repetition. So the link between cinnamon and winter does not originate in the tree's ecology. It originates in human experience layered over time. Tropical bark traveled into colder regions and became woven into seasonal traditions. Childhood exposure strengthened neural pathways connecting scent with specific times of year. And through repeated pairing, cinnamon's aroma became a quiet signal of winter, even though its roots remain in humid forests far from frost. When cinnamon is eaten or sipped in tea, its journey continues beyond scent and warmth. The volatile compounds that rise into the air can also enter the body through the digestive system. Essential oils such as cinnamaldehyde are lipophilic, meaning they dissolve more readily in fats than in water. This property allows them to pass through biological membranes relatively easily. After cinnamon is consumed, small amounts of its aromatic compounds are absorbed through the lining of the stomach and small intestine. Because essential oils are lightweight and chemically active, absorption can occur quickly. Once in the bloodstream, these compounds circulate briefly before reaching the liver. The liver acts as the body's primary processing center for many plant-derived molecules. Cinnamaldehyde does not remain unchanged for long. In the liver, enzymes begin to modify it through a series of chemical reactions. One common pathway involves oxidation, converting cinnamaldehyde into cinnamic acid. From there, additional enzymatic steps may attach other molecular groups that increase water solubility. This transformation allows the compound to be excreted more easily through urine. This metabolic processing is part of the body's broader system for handling foreign compounds, often referred to as xenobiotics. The liver's enzyme families, including cytochrome P450 enzymes, alter chemical structures so they can be safely eliminated. These processes are not unique to cinnamon. They apply to many plant-derived substances consumed in small amounts. Because cinnamon is typically used in culinary quantities, the total amount of cinnamaldehyde absorbed is modest. The compound enters the bloodstream, is metabolized, and leaves the body without accumulating under normal dietary use. The speed of absorption and breakdown contributes to the temporary nature of its physiological effects. Essential oils are potent in scent because they evaporate easily, but inside the body, they are handled through chemical transformation. The warmth felt in the mouth fades as receptors adapt. The molecules themselves are gradually processed and cleared. What remains is the memory of flavor rather than lingering chemical presence. So when cinnamon enters the body, it follows predictable metabolic pathways. Absorption occurs through digestive tissues. The liver modifies the compound into forms that can be excreted. The process is efficient and continuous, reflecting the body's ability to integrate small amounts of plant chemistry into its broader regulatory systems. Beyond cinnamaldehyde, cinnamon contains a range of plant compounds, often grouped under the term polyphenols. Polyphenols are naturally occurring molecules found in many fruits, vegetables, teas, and spices. They are characterized by multiple phenol structures within their chemical makeup. In plants, These compounds help defend against environmental stress, ultraviolet radiation, and microbial invasion. In laboratory research, they have drawn attention for their potential antioxidant activity. Oxidative stress refers to an imbalance between reactive oxygen species and the body's ability to neutralize them. Reactive oxygen species are byproducts of normal metabolism. They form during processes such as cellular respiration. In controlled amounts, they participate in signaling pathways. When present in excess, they can damage lipids, proteins, and DNA. Antioxidants are molecules that can donate electrons to stabilize reactive oxygen species, reducing their reactivity. Cinnamon contains several polyphenolic compounds that, in laboratory settings, demonstrate antioxidant properties. These compounds can scavenge certain free radicals in test tubes, meaning they can neutralize reactive molecules under controlled experimental conditions. Some studies using cell cultures have shown that cinnamon extracts may reduce markers of oxidative stress within those models. It is important to distinguish between laboratory findings and clinical outcomes. Many substances exhibit antioxidant activity in vitro, meaning in a controlled laboratory environment outside a living organism. The human body is more complex. Once consumed, polyphenols are absorbed, metabolized, and altered. Their activity may change as they pass through digestive and hepatic pathways. The concentration reaching tissues is often far lower than what is tested in isolated experiments. That said, spices contribute to overall dietary diversity of plant compounds. When cinnamon is part of a varied diet that includes fruits, vegetables, and whole foods, it adds to the pool of phytochemicals consumed. These compounds may interact in subtle ways with metabolic processes. However, cinnamon alone is not considered a primary source of antioxidants compared to larger food groups. Its contribution is modest, but measurable in laboratory analysis. Some research has explored whether cinnamon supplementation might influence oxidative stress markers in humans. Results remain mixed. Variations in dosage, study duration, and participant health make broad conclusions difficult. In some trials, slight improvements in certain blood markers were observed. In others, no significant change was detected. The evidence does not suggest dramatic effects under normal culinary use. Polyphenols in cinnamon include compounds such as proanthocyanidins, which are also found in berries and cocoa. These molecules contribute to the spice's astringency and complexity. Their structure allows them to interact with reactive species under laboratory conditions. Within the plant, they serve as protective agents. Within the human body, their behavior depends on absorption efficiency and metabolic transformation. The broader context of antioxidant research emphasizes balance. The body maintains its own antioxidant systems, including enzymes like superoxide dismutase and catalase. Dietary antioxidants may complement these systems, but they do not replace them. Cinnamon's polyphenols become part of a dynamic metabolic environment rather than acting independently. So when cinnamon is described as containing antioxidants, the statement refers to measurable chemical properties observed in laboratory analysis. These properties demonstrate the ability of certain compounds to neutralize reactive molecules under specific conditions. In practical dietary use, cinnamon contributes small amounts of these plant compounds. The effect is quiet and incremental. fitting within the larger complexity of human metabolism rather than standing apart from it. The journey from tropical forest to kitchen shelf is long, yet the physical transformation of cinnamon is remarkably simple. It begins with harvesting young shoots from evergreen trees in humid climates. The outer bark is scraped away, and the inner layer is peeled in thin strips. These strips are left to dry slowly in shaded, well-ventilated air. As moisture evaporates, the bark curls inward, forming quills. From that point forward, preservation depends on dryness and protection from light and heat. Drying is the most critical stage. Water supports microbial growth and accelerates enzymatic breakdown. By reducing moisture content, producers create a stable material resistant to spoilage. Once fully dried, cinnamon bark contains very little water. This low moisture level is one reason it can endure long transport and extended storage without visible decay. After curing, quills may be cut into smaller sticks or ground into powder. Grinding dramatically changes how cinnamon interacts with its environment. A whole quill protects its inner layers from air exposure. Only the outer surface interacts directly with oxygen. When ground, every fragment becomes exposed. The increase in surface area accelerates aroma release, but it also speeds chemical change. Light and heat both degrade essential oils. Cinnamaldehyde and other volatile compounds are sensitive to prolonged exposure. Heat increases the rate of evaporation and oxidation. Light, especially ultraviolet light, can trigger chemical reactions that alter molecular structure. For this reason, cinnamon is best stored in airtight containers away from direct sunlight and high temperatures. In warehouses and shipping containers, cinnamon is typically kept dry and shaded. Excess humidity during storage can lead to mold growth, even in low moisture spices. Producers monitor moisture levels carefully before packaging. Properly dried cinnamon remains stable because microorganisms cannot easily thrive in its environment. On a kitchen shelf, storage conditions vary. A tightly sealed jar in a cool cupboard will preserve aroma longer than an open container near a stove. Each time the jar is opened, fresh air enters. Oxygen interacts with exposed surfaces. Over months, aroma gradually softens as volatile oils dissipate. The spice does not become unsafe under normal conditions, but its intensity decreases. Whole sticks maintain potency longer than ground cinnamon. If broken just before use, a stick releases trapped oils that were previously shielded within layered bark. Grinding immediately before cooking preserves maximum fragrance. The difference reflects simple chemistry. Reduced exposure equals slower oxidation. Even packaging design reflects this understanding. Dark glass bottles limit light exposure. Tight-fitting lids restrict airflow. Some commercial producers flush containers with inert gas before sealing to reduce oxygen content. These steps slow chemical degradation and preserve aroma for longer periods. Despite global transport and months of storage, cinnamon's stability remains notable. Its low moisture content prevents spoilage. Its essential oils deter certain microbial growth. Its lightweight structure allows economical shipping across continents. The same qualities that supported medieval trade continue to support modern distribution. From tree to shelf, the process is shaped by evaporation, protection, and containment. Moisture leaves. Bark curls. Oxygen slowly alters exposed compounds. Light and heat influence degradation rates. Yet even after months in transit, a sealed jar can still release warmth when opened. Within that brittle brown powder or layered stick, volatile molecules remain ready to rise into the air. The forest's chemistry travels intact, carried quietly across time and distance until it meets the warmth of a kitchen once more. Across centuries and continents, cinnamon persisted. Many plants were used briefly in local traditions and then faded from wider memory. Cinnamon did not. It remained present through shifting empires, changing trade routes, and evolving cuisines. Its endurance can be traced to a combination of physical stability, potency, and transport efficiency. One reason cinnamon lasted is its shelf stability. Once dried, its moisture content becomes very low. Microorganisms require water to grow and multiply. Without sufficient moisture, bacteria and molds struggle to survive. Cinnamon's dryness acts as a natural barrier against spoilage. Unlike fresh herbs that wilt quickly or fruits that ferment, dried bark can remain intact for months or years when kept dry. Its essential oils also contribute to durability. Compounds such as cinnamaldehyde possess mild antimicrobial properties, as discussed earlier. While not strong enough to sterilize food on their own, they add another layer of protection. The spice does not easily become contaminated when stored properly. Stability allowed it to endure long voyages by sea and caravan without losing viability. Potency, in small amounts, further supported its longevity. Cinnamon's aroma is strong relative to the quantity used. A small pinch can flavor an entire dish. This efficiency made it economically valuable. Traders could transport compact bundles that carried intense scent. Weight and volume were minimal compared to bulkier goods. High value per unit weight encouraged careful preservation and wide distribution. The strength of its scent also reinforced memory. Humans remember powerful aromas. Cinnamon's distinctive profile ensured it stood apart from other spices. Even diluted in mixtures, it remained recognizable. That recognizability strengthened demand. Once people associated the scent with comfort or celebration, they sought it again. Cultural repetition reinforced its presence across generations. Transport efficiency played a role as well. Dried bark is lightweight and resilient. It does not bruise easily. It does not require refrigeration. It withstands moderate temperature changes if kept dry. These characteristics made cinnamon ideal for early trade networks where storage conditions were unpredictable. Its chemical composition did not degrade rapidly under ordinary handling. Geography contributed too. Cinnamon trees grew in specific tropical regions, which limited supply and enhanced value. Controlled production areas allowed traders and later colonial powers to regulate exports. Scarcity often increases perceived worth. Yet once shipping networks expanded, the spice became more accessible while retaining its distinct identity. Even in modern supermarkets where countless flavorings exist, cinnamon maintains a familiar place. Its aroma complements both sweet and savory foods. Its mild warmth bridges categories without overwhelming other ingredients. Versatility supports endurance. A spice confined to one narrow use might fade as tastes change. Cinnamon adapts easily. Low moisture content remains central to its survival. Dryness slows chemical reactions and prevents microbial growth. Combined with airtight storage, this quality ensures longevity on shelves. While aroma gradually softens over time due to oxidation, the spice rarely spoils outright. Stability builds trust. Consumers learn that cinnamon will remain usable long after purchase. So cinnamon endured not through novelty alone, but through practicality. It resisted decay. It required little space. It delivered strong sensory impact in small doses. It traveled well. Its chemistry remained intact across oceans and centuries. Within each brittle curl lies a combination of traits that allowed it to move from tropical grove to distant kitchen and remain there, quietly dependable. Empires shifted. Trade monopolies dissolved. Culinary fashions evolved. Yet cinnamon stayed. Its structure, scent, and stability ensured continuity. The same dried inner bark that once crossed deserts by caravan now rests in modern cupboards, still potent, still recognizable, still ready to release warmth when surface meets air. When most people think of cinnamon, they imagine a soft brown powder or a brittle stick resting in a jar. But cinnamon also exists in another form, essential oil. This oil is not simply melted spice. It is a concentrated extract produced through steam distillation, and its chemistry behaves differently from the bark used in cooking. Steam distillation begins by placing cinnamon bark or leaves into a chamber where steam passes through the plant material. Heat causes volatile compounds to evaporate along with water vapor. The vapor then cools and condenses into liquid. Because oil and water do not mix, the essential oil separates and can be collected. The result is highly concentrated. In culinary cinnamon, cinnamaldehyde is distributed throughout plant tissue, bound within fibers and cells. In essential oil, those volatile compounds are isolated. The concentration of active molecules becomes much higher per drop than in a teaspoon of powder. There is also a difference between bark oil and leaf oil. Cinnamon bark oil contains a high percentage of cinnamaldehyde, often 60 to 75% or more depending on species and processing. This gives it a strong, sharp warmth and intense aroma. Cinnamon leaf oil, by contrast, contains more eugenol, the same compound found in cloves. Leaf oil smells softer, slightly spicier, and less sweet. The difference reflects how various plant tissues allocate chemical defenses. Bark protects structural tissue. Leaves face different environmental pressures. The chemical balance shifts accordingly. Because essential oil is concentrated, it behaves differently on contact with the body. A drop placed directly on skin can cause irritation. Inhaling concentrated vapor can feel sharp or overwhelming. The mild warmth of powdered cinnamon becomes something much more intense in oil form. This contrast illustrates an important principle. Concentration changes experience. In powdered cinnamon, volatile molecules are released gradually as air moves across surfaces. In essential oil, molecules evaporate rapidly due to their purity and density. The trigeminal nerve may respond more strongly. Sensory receptors activate more intensely. The chemistry remains the same. The dose changes. Essential oils are often studied for antimicrobial or aromatic applications, but they are not equivalent to culinary use. A small amount of ground cinnamon in food delivers milligram-level exposure to cinnamaldehyde. essential oil can deliver far higher levels in a much smaller volume. This difference matters because the body processes concentrated compounds differently. Higher exposure may increase irritation or require greater metabolic processing in the liver. In normal dietary use, cinnamon remains gentle. In concentrated oil form, it becomes powerful. So while both forms originate from the same tree, their effects diverge due to extraction and concentration. The bark in a jar releases aroma slowly, buffered by plant fibers. The oil extracted through steam carries that aroma in compressed form. Two versions of the same chemistry, one diffused, one intensified. And understanding that difference helps clarify why cinnamon feels mild in baking, yet sharp and immediate when encountered as pure oil. Cinnamon's association with comfort extends beyond memory alone. Research in environmental psychology suggests that scent can subtly influence mood perception and even temperature interpretation. In controlled settings, participants exposed to warm-associated scents, such as cinnamon, vanilla, or clove, sometimes rate environments as feeling slightly warmer than they actually are. The air temperature remains unchanged, yet perception shifts. This effect appears to arise from multisensory integration. The brain combines sensory inputs into unified interpretations of surroundings. If a scent activates pathways associated with warmth through TRPA1 receptors, and if memory networks link that scent with physical warmth, the brain may adjust its internal estimate of environmental comfort. Perception is predictive. The brain constantly anticipates what sensory input means. When cinnamon is present, the system predicts warmth. The body may not heat physically, but subjective comfort can increase. Retail environments sometimes use scent intentionally. Studies in environmental design have explored how ambient aromas influence mood ratings, dwell time, and perceived atmosphere. Warm scents are often described as inviting. Cool scents are described as refreshing or energizing. The effect is subtle, not dramatic. Scent alone does not override physical discomfort, but it can shift emotional tone. The amygdala and orbitofrontal cortex participate in evaluating pleasantness. When a scent is familiar and positive, these regions show activity patterns associated with comfort rather than vigilance. Familiarity plays a crucial role here. The brain uses familiarity as a proxy for safety. Unknown stimuli require evaluation. Known stimuli can be categorized quickly. Cinnamon, when encountered repeatedly in calm settings, becomes part of a safe sensory library. Exposure can lower alertness slightly, reducing environmental scanning. This reduction in vigilance contributes to the cozy effect. There is also a learned pairing between scent and ritual. If cinnamon is frequently present during slow activities, baking, tea preparation, holiday evenings, the body may begin associating the scent with slower pacing. Later, the scent alone can cue that slower rhythm. Breathing patterns sometimes change in response to pleasant scent. Inhaling aromatic vapor can encourage deeper, slower breaths. Slow breathing influences vagal tone, which supports parasympathetic activation. The result is not sedation, but subtle settling. Importantly, these effects are context-dependent. If cinnamon were encountered in a stressful setting repeatedly, the association could shift. Scent memory is not fixed. It reflects lived experience. Still, in many cultural contexts, cinnamon has accumulated decades of positive pairing. That layering creates a shared psychological impression of warmth and safety. The tree did not evolve to create comfort. Its chemical compounds deter insects and protect bark. Yet through trade, repetition, and neural encoding, those compounds became part of human emotional landscapes. And so, a tropical plant, grown in humid forests far from frost, can make a winter room feel warmer, not by raising temperature, but by shifting perception. A molecule meets a receptor, a memory activates. The room remains the same, but the mind softens slightly. And that softness is part of why cinnamon endures. We began with a simple curl of bark, a dry spiral resting quietly on a shelf. Inside it, plant fibers once carried sugars through a living tree rooted in humid soil. Leaves gathered sunlight. Bark thickened in tropical air. Essential oils formed as part of the tree's own protective chemistry. Harvesting removed the outer layer. The inner bark was peeled and left to dry. Moisture evaporated. Fibers tightened. The strip curled inward, shaped by physics rather than design. What remained was light, brittle, and shelf-stable. Within that curled bark, cinnamaldehyde waited, a small molecule capable of drifting into air, binding to receptors, and creating the perception of warmth. Not heat in temperature, but warmth in sensation. TRPA1 receptors activated. The trigeminal nerve carried its quiet signal. The brain interpreted it as gentle internal glow. We followed cinnamon across trade routes and oceans, from Sri Lankan groves to Egyptian temples, from Roman markets to medieval ships, from colonial ports to modern cupboards. Its chemistry remained unchanged while the world around it shifted. We explored how scent connects directly to memory, how familiarity reduces vigilance, how childhood pairings can shape lifelong associations. A tropical bark became linked with winter not because of climate, but because of repetition. We looked at how the body processes its compounds, how the liver transforms cinnamaldehyde, how polyphenols behave in laboratory studies, how essential oils differ from culinary spice. Concentration changing experience. We noticed texture, brittleness, the way grinding increases surface area, the way light and heat slowly soften aroma over time. And through it all, cinnamon remained what it has always been. Dried inner bark of an evergreen tree. A plant defense compound became a human comfort signal. A volatile molecule became part of ritual. A low moisture bark became one of the most transportable spices in history. Now, if there is cinnamon nearby, you might notice it differently. Not just as scent, but as structure. Not just as warmth, but as perception. Not just as flavor, but as chemistry meeting memory. The room remains the same temperature. The air moves quietly. Perhaps there is a faint trace of spice somewhere in the background. And if there is, you may notice how gently it rises, how quickly it fades, how easily it blends into stillness. The bark, once dried in tropical shade, now rests in calm air. And so we settle here, in that same quiet warmth. Thank you for spending this time exploring the science of cinnamon. Rest well.